Let's talk about DNA. I mean, really think about it. It's the blueprint for every single living thing, right? A microscopic code that decides pretty much everything about us. But here's the crazy part. It's written in a molecule so impossibly tiny, you could never ever hope to see it, let alone edit it by hand. And yet, that's exactly what scientists have been doing for decades, literally cutting, pasting, and copying the fundamental instructions of life itself.
So, how in the world do you do that? How do you perform surgery on something you can't even see? This isn't just a tough question, it's one of the biggest engineering challenges in all of biology. You can't use scalpels or tweezers, you need tools that are infinitely smaller and way, way more precise.
The answer is you need a toolkit made of nature's own microscopic machines, enzymes. These are incredible little proteins that make all the essential reactions of life happen. And for molecular biologists, they are the absolute must-have instruments for rewriting DNA. Okay, let's actually dive into this toolkit. We're going to open it up and take a look at the three cornerstone tools that didn't just move biology forward, they completely changed the game. We're talking about the scissors, the glue, and the copiers. First up, the molecular scissors. Look, before you can build anything new, you got to be able to make a clean cut. And in genetics, that means snipping the DNA double helix, not just anywhere, but at a very specific predetermined spot. The tool for this job is called a restriction enzyme. Just think of it like a pair of programmable biological scissors. It's an endonuclease, which just means it cuts inside the DNA strand. But the key is, it doesn't cut randomly. Oh no. It is programmed to recognize one very specific sequence of genetic letters out of a genome that might be billions of letters long. And how they work is just elegant. These enzymes basically patrol the DNA helix, just scanning the code over and over. The second they find their exact target sequence, and only then, they lock on and make a perfect cut. This single ability is what lets scientists create manageable DNA fragments, basically pulling a single gene out of an entire genetic library. Now, what's really amazing when you look at this timeline is just how fast this discovery changed everything. It all started back in the early 50s with a simple observation that some bacteria could fight off viruses. By 1970, we'd isolated the specific enzyme doing the work. Just 8 years after that, it wins a Nobel Prize. And by the early 90s, this thing that started as a curiosity in bacteria became a cornerstone of forensic science. I mean, that's an incredible pace from basic science to a world-changing technology. So you've used your molecular scissors and you've made the perfect cut. Great. What's next? Well, you need a way to paste that DNA fragment somewhere new. And that brings us to the second essential tool in our kit, the molecular glue. This biological superglue is an enzyme called DNA ligase. Inside our cells, its normal job is to repair any breaks in the DNA backbone, but in the lab, we can hijack it to permanently join two separate pieces of DNA together. It creates the strong chemical link, a phosphodiester bond, and literally stitches the helix back together, making two pieces become one. So, here's the crucial part. This pasting process, which we call ligation, is the absolute heart of genetic engineering. It's the step that lets a scientist take a gene from one organism, say a human, and seamlessly stick it into the DNA of something else, like a bacterium. This is how we create what's called recombinant DNA.
And this is the perfect example of how powerful it is when you combine the scissors and the glue. One of the first huge wins for this technology, way back in 1979, was making human insulin for people with diabetes. Scientists used the scissors, the restriction enzymes, to snip out the human gene for insulin. Then they used the exact same enzyme to cut open a little circle of bacterial DNA. Then in comes the glue, DNA ligase, to paste the human gene into the bacterial DNA. And the result? The bacteria, now carrying human instructions, turned into these tiny little factories, just churning out life-saving insulin.
All right, let's move on to our third and final essential tool. So you've cut the gene you want, you've pasted it into place. But here's the problem. You're often starting with a tiny, tiny amount of DNA. To do anything useful with it, you need more, a lot more. You need a molecular copier. So our toolkit has two main types of copiers. The real workhorse is DNA polymerase. This is the enzyme that reads a strand of DNA and builds its perfect matching partner. It's the engine of all DNA replication. But then there's this other, really special copier, reverse transcriptase. For the longest time, the rule in biology was that information only flows one way, from DNA to RNA. Well, this enzyme blew that idea out of the water.
It showed you could actually go backwards, from an RNA message back to DNA.
And this ability to copy genetic material is just fundamental. It's the power behind PCR, the polymerase chain reaction, which lets us make billions of copies from a single piece of DNA. It's absolutely essential for DNA sequencing, which is really just reading all those copies. And with reverse transcriptase, we can get a snapshot of what a cell is doing by turning its temporary RNA messages into stable DNA that we can study. This tech even fuels those rapid diagnostic tests for diseases that you can use right out in the field.
So when you put it all together, the scissors, the glue, and the copiers, you realize these three tools built the entire foundation of modern genetic engineering. They give us the power to read, write, and edit the code of life itself. But, like any good toolkit, this one is always getting upgraded. This quote from the source material really just says it all. The ability to just cut DNA in a predictable way wasn't just another small step. It was a turning point that changed the entire direction of science and everything that came after it. And that brings us to the next generation, most famously CRISPR-Cas9. The key innovation here is really clever. With an old-school restriction enzyme, the target it cuts is hardwired into its physical shape. If you want to cut a different DNA sequence, you have to go find a whole new enzyme. CRISPR, on the other hand, separates the scissor from the guide. The Cas9 protein still does the cutting, but it's told where to go by a little easy-to-make guide RNA. So if you want to change the target, you just make a new guide. It's the ultimate upgrade, a truly programmable molecular scissor. But here's the key thing to remember. This isn't magic, it's evolution. CRISPR is built on the exact same fundamental principle we've been talking about, making a precise cut in a strand of DNA. It's just a vastly more flexible and powerful way to do it, but it's standing on the foundation laid by those first molecular scissors decades ago. So from those first kind of clunky restriction enzymes to the sleek, programmable gene editors we have today, the DNA Architect's Toolkit has become more powerful than anyone could have possibly imagined. And it leaves us with this one kind of awe-inspiring question to think about. As these tools keep getting sharper and better, what are we going to build next?